Hey everyone, welcome to Foresight's Neurotech group. Today we're really lucky to have Gerald Pao joining us all the way from Japan from the Okinawa Science and Tech Institute. And today he'll be talking about algorithms to map neural activity to behavior. I was looking at your bio, Gerald, and it's so interesting. It looks like you've been everywhere and have done everything. So we're really lucky to have you on and excited for the presentation.
Oh, thank you. All right, so today I'm going to talk about basically how to generically map algorithms to generically map brain activity to behavior. And we're doing this in the framework that we work with is basically on manifolds. So what is a manifold? So basically, if you look at these three ordinary differential equations, they make up something called, that's very well known, like the Lorentz attractor. And it's made up from three ODEs. So each equation, like x depends on y and x, y depends on x, y, and z depends on x, y, and z, as this formula shows. But if you look at the manifold, this is the relationship between those three variables, and they progress over time. And if you see that, for example, on the left side here, when the values of x are low, then z and x are negatively correlated, but z and x are positively correlated on the other side. And if you look at a single variable as basically nothing but a time series as a projection, if you move time forward, is the projection of the shape as this thing evolves over time. And you can project also y and z. And if you actually take the three time series and then plot them against each other in this phase space, then you get the manifold. So you can interchangeably go between the manifold to basically to a time series, and you can also go from time series to manifold. So both are interchangeably, you can go from one to the other. So we actually use these objects, these mathematical objects, these shapes, which we call attractors or manifolds. And there was a remarkable discovery with them back in 1978, but it was published a paper basically in 1980 of three graduate students and undergraduate without a professor. They actually found that you can actually take a single time series and convert it into a shape. So this is basically this paper called Geometry from a Time Series. And that was essentially what later became known as the Tokens theorem. So the Tokens theorem was named Tokens because although these guys discovered this technique of how to convert a time series into a shape, they didn't get much credit for it because in mathematics, if you don't have the proof, it doesn't count. So that's why I call Tokens theorem, because Tokens in 1981 came up with a proof as a part of the meeting report for this thing to be generically true. So the proof of Tokens is the one that people consider important. So what does the Tokens theorem tell you what you can do? Tokens theorem is basically if you take a single time series, let's say in the three variables that you have is x, y, and z, and let's say that you don't know what y and z have, and you can only have measurements of x. If you only have x, what you can do, you can still take the time series of x and make a shape out of it. And the way you can actually make a geometric object out of it is by taking the time series of x, then you shift that same time series over by this amount tau, and then shift that amount by 2 tau, and make this your new x, y, and z. If you do that, you get this shape here, which is called a delay embedding or a shadow manifold. This is a shadow manifold of x. And this is basically a stretched out form of this one. So this is called a diffeomorphism. A diffeomorphism is like a stretched form of the original one. But one thing that is remarkable is, for example, for any point that's on this, you can see that there's two lobes. The two lobes are preserved, so the topology is preserved. And there's basically only the distances between the nearest points are altered, but the identity of the nearest points for any point on the manifold are preserved. So this is essentially say, if you only observe one component out of a thing that has many components, you can still know something about the other components, that they exist, and which ones they are, you don't know. They exist, and you can estimate how many. And you can actually use this to predict the future states of this thing. I'm just going to briefly show a video here. that just states the thing that I explained to you. Since this is super important, I'm just going to repeat this with this video. And this is the Takens theorem. You have the 3 time series here. And the 3 time series that you actually hear are actually identical to each other. One is x of t is xt minus tau, shifted over by this amount tau, and then shifted over by amount 2 tau. So once I actually start plotting those against each other, you're going to see that you actually start generating a manifold that's called the shadow manifold of X. And you're going to see how this changes over time. So as this value changes here, you plot it on this, and that's basically what you get. So this is called a delay embedding or a shadow manifold. So what kind of things can you do with this? So here, for example, we have a time series, and we want to know if this time series is chaos or noise. So if it's noise, means it's intrinsically not predictable. If it's chaos, means that you can do short-term predictions, like you can predict weather. You cannot predict weather like a year in advance or something like that, but you can predict weather like a day or 2 or even 9 days or 10 days in advance. It becomes less accurate the farther out you go, but you have some predictability as opposed to no predictability if the thing was completely random. So how do we know if this is predictable or not? So here we can actually show you a delay embedding. So let's say you have these two. I'm already telling you which one is chaos and which one is noise. If you take let's say the first value, we call this today. And the second value, the second time point, we call it tomorrow. Then we can make a 2-dimensional vector. We can just use X and Y as coordinates, and we can plot it in phase space like this point here. Then we can actually go to time point 2, make that X, and time point 3, make it that Y, and then plot the next one. And if you do this iteratively over the entire time series, you can see over time that you actually get certain levels of structure in chaos, but in noise, you see no patterns whatsoever. So let's say I know the value today of X is -1, then you can predict that the value of tomorrow is going to be about 0.75. Just read it off here, right? So you go here, you go up here, you hit this, and then you can see it's 0.75. But certain points here are ambiguous. For example, if I key the value today now is 0.75, there are all these other values that you could possibly have. So your prediction is going to be inaccurate because these things are stacked over each other. So they're non-unique. This is what you call singularity. The trajectories are crossing. But what you actually can do is basically you can give this make it a little bit more complicated. I'm not going to take the value today, the value tomorrow. I'm going to actually take yesterday, today, and tomorrow. If you actually put it in 3 dimensions instead of 2, now all the trajectories become unique. Their things don't stack anymore. So if I know 2 of them, I can tell you what the value of the third one is. So this is basically, in 3 dimensions, it became highly predictable. So if we actually generalize this thing, is that how does it mean that we can actually use this embedding to make short-term forecasts? So what do you do here? Let's say you have a particular point. And then you find a point here, and then you find its nearest neighbors. What's it going to do in the future? You take the neighbors that you have, each of them also have a time index, basically a time stamp of when they were taken. You project it forward 1 time step, or 2 time steps, or 3 time steps, and then the center of that simplex is where you're going to predict what the future state is going to be. And this way, you can actually predict things into the future. So you can actually predict into the future, even if you have only a single variable, you can actually get the other variables accounted for as delays. Those are the other mystery variables that you actually had. You know exactly what they have, but you approximately know what their behavior is. So typically what we do to test these models is basically we take half the time series, we make it to a library, that we make this delay embedding, and then we'll try to predict the second half. And then we see how well do we do. So if you actually do this, you can actually see that, for example, in this particular case, with 2 dimensions, you actually get this line, and then 3 dimensions, it becomes better. In 4 dimensions, it's still about the same, and then it starts degrading. So Betsy, why does this happen? Why does it become worse with more delays? So what happens is that you start adding things that are irrelevant. What do I mean with it? Let's say, imagine that you're trying to predict the weather. If you want to predict the weather, if you take information from the previous day about the weather, maybe it helps you to predict the weather tomorrow. Maybe it helps you also to take the day before yesterday, not yesterday, but also the day before yesterday. It could be also really relevant to help you predict the weather tomorrow. It has some property that helps you predict. But if you take weather from, let's say, 2 weeks ago, that's completely irrelevant, then you're diluting the good information with noise. And that information, whether from 2 weeks ago, is completely irrelevant. So then you're diluting your good information with the bad information. And then your predictability, the accuracy of prediction decreases. So this predictability is basically the correlation coefficient of observed versus predicted. So this is the mean over all the predictions of observed over predicted. The degradation that you show here is that you're diluting the good information with the bad information. And then you can, if you do this, you can actually determine the system is probably around, about 3 dimensions or so. This is how you can actually estimate how many variables actually the system had. So even though you don't know what the other variables are, you can estimate the number of variables. Anyway, so you can actually, so in 2011, so that in original proof of tokens was done in 1981. In 2011, there was an extension of the tokens theorem. And this extension of the tokens theorem tells you that actually you can mix, delays with real variables indistinctly. So for example, here you can actually see we take the real variable z, the real variable y, and we take a delay of y, and it's also still diffeomorphic. It's basically still a stretch form of the original one. It's just stretched in a different way. When you just take y, you have stretch from this one as a different stretch, but you can actually see that it's still diffeomorphic if you mix real variables and delays. This becomes important because in 2012, there was a paper in Science that showed, and this is a paper in theoretical ecology and quantitative ecology, that show you generically there's a method that was invented by George Sugihara, and this method is called convergent cross-mapping. And this method allows you to take time series and using Tokens theorem to find cause and effect relationship between time series. So how does this actually work? So the principle is simple. So as I showed you earlier, this is the original Lorentz attractor, and this is the delay embedding of X. So each of those points exists on the other one. So it has a one-to-one mapping of the other one. It's the stretch form of each other. So if it's stretch form of the other one, that means that every nearest neighbor that exists here is going to be the same nearest neighbor for the original one. But you can also do it for y. So if y would also have a nearest neighbor, and they would be the same ones, equivalent, but they're just in a slightly different position because they're stretch form of each other. But if they shared if every point shares all the nearest neighbors, then that means that they originally belong to the same system. So if you see that X and Y share all the nearest neighbors, and then you can use this to predict across each other. So I can use X to predict Y and Y to predict X in this particular case. If they can do this cross prediction, this basically predicting across variables. If you can predict across variables, that is evidence that originally they belong to the same system that had X, Y, and Z. You don't know Z, but at least X and Y, they belong to the same system. And then you can maybe use a delay of X or delay of Y to make it 3-dimensional. And then at least you know that X and Y together belong to the same system. So this is the method. It's called convergent cross-mapping. It's very successful. And it's predicated on the idea that if two variables originally belong to the same system, they're also going to be stretch forms of each other if you took the single variable delay embedding or shadow manifold. And this works because the information that is shared between them allows you to predict across each other. So basically, if they share information, they are also causally related. Okay. So this is basically the method called convergent cross-mapping, and that was invented in 2012. But there is one more thing is basically if you take the nearest neighbors, the more data that you have, the nearest neighbors become more dense and closer to each other, so your predictions become more accurate. So basically, with more data, L tells you the amount of data they're using. The more data that you have, basically, the better you can predict. And this row here is observed versus predicted mean over all the predictions that you do. And then you can see that if you converge, meaning with more data, it becomes more accurate. That is another criterion that is necessary to show that that CCM is actually, that you actually have real causality. That's not just by chance, that you actually have some similarity. Of course, in the real case, nothing is perfect, but here I'll show you an example of what you can do with this in neuroscience. So in this particular case, we actually had basically this area of the brain called the lateral parabrachial nucleus. We actually had calcium signals. This is GCAM that you actually expressed in there with the virus. And then you get a calcium signal that gives you a neural activity of this area. And then you use an optical fiber to measure that. Basically, you stimulate and look at the fluorescence of this area, and then you get a calcium trace of this, or the fluorescence. At the same time, you actually put a platinum wire into the nose of you implant a platinum wire into the nose cavity of the mouse. And when the mouse breathes out, warm air gets onto that thermistor, which is the platinum wire, and then the resistance goes up, and you can measure the resistance, the electrical resistance. When it breathes cold air in, the temperature drops on the wire, and then the resistance goes down, and then the voltage changes. And then you can get a voltage change for every breath the mouse takes. So you can calculate the breathing rate of the mouse. If you try to correlate these two variables, the best that you can do is less than 0.3. But if you actually time shift it, because the breathing rate and the brain activity are a little bit delayed with the researcher, if you just time shift it to match it as much as you can get almost to 0.4, but not quite. So that's the level of prediction that you can actually achieve with correlation by using a linear model. However, if we make this into a manifold, and we make this into using delay embedding, make it into a manifold to predict each other, we can actually use this using CCM and the prediction. You can see the predictions become really good. You actually can do 97% accuracy in your predictions at 9 seconds out. So you went from no instantaneous, like at time zero predictions at maximum 0.4 to 97, with 40% accuracy to 97% accuracy 9 seconds out. For breathing, that's an eternity. And what you can see is that, that the calcium causes the breathing, but then also the breathing can control the brain activity. And everybody who has done yoga knows that if you actually slow down your breathing, and also when people tell you not to panic, you can actually slow down your breathing and calm down. You can actually see this effect in the calcium affecting the breathing too. So the brain activity can affect the breathing, and the breathing can also affect the brain activity. So as a feedback loop, and you can actually detect it here. So we can actually experimentally test if this is real. So for example, here, this brain area is actually sensitive to opium. So if you give it an opiate, so that you can, if we give it like saline, which is just 0.9% sodium chloride solution, nothing happens. The observed and predicted are very accurate. But if you actually give it morphine and look at the time series, you can no longer predict. Your prediction accuracy becomes very low. So basically, those predictions from CCM are experimentally testable. It's not just that you can test the thing, you can predict things. You can actually show that the predictability goes away if you do an experimental manipulation that affects the cause and effect relationship of these two things. So this is like experimental proof of your computational hypothesis. So can we scale it up? So this is basically, we can actually look at the brain of zebrafish, and we put basically a calcium indicator that measures the levels of calcium in every single nucleus of a neuron in the zebrafish. And you can have about 120,000 neurons that you can measure simultaneously with light sheet microscopy. And that's what you're looking at here. The entire brain at single neural resolution. And we put into the nucleus in order to be able to segment. So every dot is a nucleus. It separates out between the different neurons. So here you're looking at the whole brain. Can we understand this? If you actually do this, if you actually look at this, when I started doing this, I only have 70,000 neurons. If I'm going to calculate this, it was going to take me 4,600 years to calculate. So obviously, this is not something that's doable because the thing was like, so we modified this. The first thing we did was brute force. We used number 5 supercomputer in the world at the time. This was in 2018, brand new supercomputer at the time, was number 5 in the world. And we ran this, and it took basically 8.5 hours with 512 nodes. But now we actually improved the software with a whole bunch of changes. And now we can actually do the whole thing in 20 minutes in a single node. So from the beginning of the project, through software improvement and also hardware improvement, we actually got 122 million times faster. The algorithm runs at 122 million times faster. So now it's actually at a level that we can actually use it for brain scale kind of problems and routinely. Okay, so what kind of things can you find out with this? For example, you can actually see, because remember, we can actually estimate the dimensionality for each neuron. So what you actually see here is how many neurons have the dimension 1, how many neurons have dimension 2, how many neurons have dimension 3, and you can actually see the distribution of dimensionality across neurons in the brain of a zebrafish when it's trying to, when it's under its normal conditions or when it's trying to escape from low oxygen. So we actually bubbled nitrogen into water, eliminated a lot of the oxygen, and then passed the water through the fish that we actually image from, the zebrafish larvae that we image from, to be exposed to this low oxygen water. And the first thing they want to do is try to escape this low oxygen condition. And then you actually see what it is. So what you actually see that when it induces the escape behavior, then this high dimensional stuff kind of goes down and shifts everything to the left. So the dimensionality decreases when the fish, because it actually makes sense because, for example, if the fish, and where do these things happen? So if you actually look at the normoxia versus the hypoxia condition for the fish, and you look at the dimensionality that we measure for each neuron that we actually have there, you can see this yellowing part. That means that the dimension goes down. The lower dimensionality happens in these motor areas, in the midbrain motor areas especially. But in the visual tectum, it doesn't change because the visual tectum is receiving, basically, it's like the V1 in mammalian cells. You are receiving the signals that you actually have from the environment. And as complicated as the environment is, that's basically the dimensionality that you're going to see. But as the fish was doing a whole bunch of different movements and behaviors, but when it gets exposed to low oxygen, all that it wants to do is escape. It only tries to get away. So you reduce the complexity of the behavior, and then the dimensionality actually goes down. And that's basically what you're seeing. So you can actually see where in the brain that dimensionality goes down. So even if you have all these interactions here, how do we understand this complex? This is the cause and effect matrix of every neuron to every neuron, how strong the causal relationships are between them. But how do we make sense of this whole thing? It's too much. It's like there's, even in the case of a zebrafish, you have 2 to the 8 and 10 to the 9 interactions here, and you're none the wiser by looking at this thing because just trying to drink from a fire hydrant, right? Okay, so we had to come up with a method to make sense out of the things. So what we actually want to look at is basically these nonlinear interactions. What do we mean by nonlinear interactions? Basically, we want to say, where are the decision-making places in the brain? And the way we do this is by taking the idea of taking CCM, which is this convergent cross-mapping result, that is basically, this is causal things, and then subtract from it correlation. Correlation is the stuff that's basically redundant. If it's highly correlated, it means the same. So we just see that where does CCM do better than correlation? We subtract one from the other one. And here, for example, you can see basically you have 154 representative classes of neurons. So basically each of these neurons for you to see more. In the case of normoxia, you have many different clusters, and then it becomes more homogeneous under the low oxygen, because before you actually could have many different interactions between the neurons because you had many different behaviors. But as you decrease the oxygen and want to escape, then it becomes much more homogeneous. Okay. Then if you do this basically subtraction that we call causal compression, we subtract the CCM predictability from the correlation, you actually get these residuals. And these residuals means that, for example, this thing, which is, for example, the fish swim behavior, is determined by these neurons. So this guy is integrating information from us. So this is the effect, and these are the causes. So the causes are the rows, the effects are the columns. So this is basically this neuron here is integrating non-redundant information from all these rows. So what does this actually mean? So in this way, we can actually summarize the behavior of the entire fish into 3 neurons. And here we actually essentially did dimensionalize. So we actually have 3 representative neurons here, and this is the escape behavior that you actually see here. And if you actually make it into this manifold that you see here, the manifold on the right side, and if you, let's say, make a little mask around here, every time that you see one of these bouts, basically use a fish swim bout of escape behavior. It's basically a fish turn. And each of the swim bouts, so basically we can actually, if we use this map, we can actually predict at least half a second ahead when the fish is going to make a bout and what type of bout or swim bout is going to make. So in the end, we actually reduce the decision-making space of those 70,000 neurons down to 3 neurons to explain this into something that allows us to explain the decision-making of the fish in terms of things that are non-redundant and allow you to predict. Okay, so then I actually have to go to this idea. What is this idea based on? So the idea is based on this relationship between correlation and causation. And typically we think of correlation, we look for things that are correlated, and within those, we actually do an experiment to see if they're actually causal, because everybody knows that correlation does not guarantee causation, right? But we usually never think of causation without correlation. And that's something very unusual to think about, especially in biology, where people always try to start with a correlation. So here I'll show you an example of this, why the typical things that we do when we do statistics and correlation, we're actually doing. So here I actually have these 2 movies of these conditions. There are 2 conditions and where I used to live in San Diego. And then I'm trying to compare these 2 things. And what people typically do says, okay, I want to compare condition 1 with the Garibaldi fish and with the sea lion. And if I want to compare the 2, if I'm a rigorous scientist, what I'm going to do is I'm going to take 10 repeated samples of this and 10 samples of this, and then average and determine mean and standard deviation, and then compare the 2. So if I take 10 random frames of this movie and 10 random frames of that movie, overlay them all, and then compare the differences, this is what I'm seeing. Here, basically, what you actually see is that, yes, we have some differences, a little bit of orange here, a little bit browner here, and a little bit more green there, but it's missing the point. Because if you use statistics and you use basically averaging, every fish that's not in there, you're going to consider noise. And if you have a system that's dynamical and you apply statistical methods that are static, meaning that they assume that the things are in equilibrium, then this is what you're doing in the model. So obviously, this is not ideal. So how do we avoid this? So one way to avoid this is to take time series. So we look at the things, each observation are like parts of a frame of a movie. We take time series. So if you take time series, then you can actually look at things as if they were frames of a movie. So here, actually, I show you 3 genes, not neurons, but bear with me. It doesn't really matter what it is. There are 3 genes that we actually collected data for all through the cell cycle. And these 3 genes are all uncorrelated. They have correlation coefficients of less than 10%. So if you actually like 2 of them, these 2 genes, switch4 and CLIN3, the correlation coefficient is 0.08. And people would actually, if you do the linear regression, there's essentially nothing there. You'd say there's nothing going on there. But if I say, since these were time series, I can actually give you this was which one was time point 1, which was a time point 2. I can take time point 1, take a line and put time point 2, time point 3. And if you actually see, there's actually something that is very coherent, and across 2 replicates of the cell cycle, it actually repeats. So there's a relationship here. It's just that it's not a straight line. It's not based on linear correlation. You actually have basically structure here. But as before, here some trajectories cross, and these are ambiguous. So if I were to know that this point of, the levels of CLIN3 is 40, I can tell you the level of switch 4 should be around 2. And if the level of CLIN3 is about 30, it should be about 4. But when I get here, if the level of CLIN3 is about 45 or so, there are all these possible values, and this is ambiguous because it could be any of these. So how do I solve this? I actually increase the dimension. So if I take a 3rd gene and add it onto it, now the trajectories become super clean. There's no ambiguity. And now you can actually predict into the future, because if you're here, you can predict what the next step is and just walk along the shape that you actually have. This is your manifold of these 3 genes. They have no correlation with each other. They all have less than 10% correlation, but they seem to form a coherent structure, and they have a relationship. It's just not based on correlation. So actually, when we calculated this, we actually found 4 genes were part of this network, and this will be calculated by CCM that they were related. So I put here these 4 genes. Since I cannot put in 4 dimensions a picture in your 2-dimensional screen, this is basically the 3 dimensions. Each of them is 1 gene, and the 4th gene I show you as the intensity of red. So I have this as 4-dimensional manifold, and then the intensity of color there is the 4th gene. Okay. So this looks all nice. It looks very elegant and like satisfying. But how do I know this is real and it's not a mathematical fantasy of mine? So I just found these things, but how do I know this is real? So the way to figure out if it's real is maybe do an experiment to prove it. And how would I prove it? So these things I'm saying they have no correlation to each other. So they have no correlation to each other, like less than 10% correlation to each other. But if I were to do an experiment, if I say alter one of them and the other ones respond to it and change their trajectory, that would indeed be an experimental proof of causation without correlation. And if you look at here, the blue line is when I overexpress. I make like high amounts of the red gene, and the response of the other genes is what is the blue line. So the trajectories change. This is an experimental proof that there's causation without correlation, because there's cause and effect. The other one responded. When I overexpress V5, the 3 other genes basically altered their levels of expression, altered the behavior in response to overexpressing V5, although they had no correlation to each other. So this is basically, if they were not related to each other, if they were not causally related to each other, they should not change their trajectories at all, right? So this is basically what it is, experimental proof of this. So what does this actually mean to have causation without correlation? And most of us, we're not accustomed to thinking of it, but I'll give you like a everyday example of causation without correlation that is going to make the idea a little bit clearer. So let's say that I make a rule for myself. When do I drink red wine? And I say that I will drink red wine because I'm not a drunk. I only drink in the evenings. And then I will only drink with cheese and steak for dinner. But I will not drink in the mornings or lunch, but only in the presence of both wine and cheese. If I were to look at a time series of the presence of the appearance of steak and cheese and the time of the day, in my week. So when cheese appears for breakfast and for lunch, I don't drink. When steak appears and for dinner, but there is no cheese, I also don't drink. And if you look at the appearance of cheese, this basically the time that it coincided with red wine drinking, it's only 1/12. 1 in 12 occurrences was cheese and steak, and also in the evening. So if you actually require multiple conditions to be satisfied, when it's basically you need a perfect storm, frequently each of the individual variables do not correlate with the outcome that basically that you're trying to find. If there's information integration of multiple things and multiple conditions that you could put together as a Boolean of and, or, etc., then you can actually get causation without correlation. So causation without correlation gives you the rule making. When you look at things that are correlated, basically you're looking at the bucket brigade. If I pass, a bucket brigade, the same information to the next step and nothing gets lost, then it will be highly correlated. But if you look at the decision-making places, you're going to have causation without correlation. Anyway, so how do we formalize this? The way we formalize it is actually into a method. And the method we call causal compression, where you eliminate the things that are not, you use CCM to find the things that are causal. You eliminate the things that are correlated, and you look for the things that are causal but uncorrelated. Then you can actually summarize the entire system in terms of the things that are causal but uncorrelated to your target variable that you care about. And then you actually take each of the axes correspond to a particular neuron that's good predicting your stuff. And then you can actually add the redundant variables to get the variety that you can get. But each of these things are completely identifiable because each of the axes that I show you here correspond to particular neurons and particular brain areas of particular observable, and they're not what you call a latent variable. So it's not like a PCA. If you take a PCA, your PC is like 70% neuron 1, 20% neuron 2, 30%, like it's a projection. It's a mathematical tool that you use, but it doesn't have any direct correspondence. Each of your PCs doesn't have a direct correspondence to something in the real world. But if you don't have correspondence to the real world, you cannot do an experiment on it. On the other hand, what I show you here, each of these things corresponds to a particular neuron or a particular observable, or like a brain area, that you can tell you where should you do an experiment to try to test if this cause and effect relationship is actually true. So if you actually use causal compression and predict things, we predict things fairly well. So here, basically, we actually try to predict the forward speed. We just make these manifolds based on 5 different brain areas in a fly that's like walking on a styrofoam ball where we record calcium. And you can see that you actually predict quite well with causal compression. That's the blue line. That's observed versus the real values is the forward speed of the fly is the black line. And then you can see that we do better than PCA. And then this is the forward speed and there's the left-right speed. And in both cases, you identify particular areas and they're like directly observables. And these are interpretable. So for example, like time series 26 that correspond to a particular area in the brain is like when the activity is low, the animal doesn't turn either left or right. When it reaches this intermediate level, it can turn left quickly. And then at the higher level, it can turn left quickly too. And then what turns right is preferentially only when the activity is high. So you can actually see that these manifolds that you actually generate are actually directly interpretable. You can actually know what are the conditions under which the animal does what behavior. So you can actually do this on fMRI. Here we show there's basically the standard way to predict things is using like generalized linear models, and this is the prediction accuracy. And with this multivariate GLM, you can actually only go in this range. You can actually show that with these r-dimensional reduction methods, actually you can do substantially better. Anyway, but you also identify particular regions where this happens. So here's another example. to make it a little bit more obvious, so this is a task. This is a collaboration with somebody at UCSF, Lauren Frank, and his postdoc Shi. And here basically is basically a rat that is having a maze task that has to decide into which maze it wants to do. It doesn't really matter what the task is here. And then you can actually say, I want to try to see which neurons allow me to predict where in X coordinates, like which tunnel it is in, and which neuron tells me how far along the tunnel the rat will travel. So if you actually look at X position, it's all CA1 neurons, and the one prefrontal cortex, the majority of the information is from CA1. So it tells you where the information comes from to allows you to predict the X position. The Y position, on the other hand, is from medial prefrontal cortex, orbital frontal cortex, which is the decision making and which tunnel you want to go into. So not only it allows you to basically to allow you to predict up to about 90% accuracy, it also tells you from which places the information for this decision come from. The other thing that's interesting is that you actually have, if you actually have local minimum, like you guys see, this algorithm completely allows you to climb out of it. So this one is pretty smooth, but on the other hand, predicting the Y position is much more difficult. And you can actually see that you can overcome really like these local minimum. And we can actually show that, for example, we can actually do better than LSTMs for behavior of mice. In this case, mice, we're checking whether the mice are, how much oxygen they're consuming, how much they're moving, and basically how much they're drinking, and how much food they're eating. And then, for example, this is the performance, like 80% accuracy with this MDE algorithm that we actually have. And then with LSTMs, which is your standard recurrent neural network, you can only do about 30% accuracy. Anyway, if you do this, you can actually predict, for example, we actually give the mice an injection of something called LPS that makes them very sick. This is a bacterial component that makes achy, and actually, if it's a lot, you can actually get septic shock to change a state. You can actually use the failure to predict to do something useful. You actually can use the failure to predict as a evidence for a change of state. So when we inject the animals, you can see your RMSE, your error in prediction goes up. So before you predict, if you look at observers who predicted, observed as blue, predicted as orange, and then see we do fairly well. And then you do this injection, the mouse is fundamentally feeling different. And then you can actually see the oxygen consumption goes down and your prediction error becomes very big. You can actually see when the mouse gets sick, the prediction error becomes big and it starts to recover. Then the prediction error becomes small. You can use this as a method for anomaly detection. So you can define what's normal from what's abnormal, and then you can use this for anomaly detection of a system that you're interested in. Okay. I think I'm running out of time, so I'm just going to skip over a few things to go to the final thing here. So this will be like, how do we actually use this framework to download the brain into a computer? And basically what we're doing is like we're using CCM, as I showed you before, in this case, the fly. The fly, basically you look at neural activity and you also record the movements. And then basically we use this causal compression business or MDE to basically only focus on the decision-making neurons. And then basically we make a network of manifolds. And what do we mean by this? Let's say you want to predict the left and right movements of the fly. And then just for argument's sake, we take the neuron that helps us to make a three-dimensional manifold to predict left and forward. But we have two unknowns. This is something that we cannot deal with. So here we want to predict left, but if we want to have forward and left, right at the same time, we can't have it because we have two unknowns. One way to overcome this is that we're going to make a manifold that specifically tries to predict forward. And then we place this forward value, we pass it on to the next manifold to predict. And then we only have one unknown now, that we can actually have forward and left, right. We pass the information forward and this. In principle, you can do this for every time series. And then you can link the entire network up. Basically, you can predict every time series that belongs to another manifold with another manifold, and then you make a manifold, a network of manifolds. And then this network of manifolds basically can predict each other, and finally predict like the forward and left right of the fly. And then you can also accept that the prediction is true, so you can run it forward. You're accepting the next prediction and the prediction after that, and so on and so forth. You can keep running it forward. If you do this, this is the training set of forward speed of the fly. You can see that the fly runs fast and then slows down, runs fast, it has these bouts. If you run in the computer forward, you can actually get pretty realistic looking bouts of behavior in running it forward. And this also happens for neural activity. This is a neural activity as it recorded. You simulate it forward. It actually makes something pretty realistic. And if you look at the spectrum of the forward movement of the real one versus the generated, the correlation coefficient of the two is 92%. So it's not just me by eye saying they're similar. If you look at the spectrum is like 92% similar. If you look at, for example, the left-right speed of the fly and the forward speed of the fly, so the fly is basically like sitting still, it'll be here, runs fast forward to the left will be here, runs forward to the right will be here. And if you follow the whole through, this is basically the behaviors that the fly did. This is the simulated fly. But if you leave out the brain areas, basically, maybe the fly is a very simple animal. So if every step allows you to predict the next step, if you try to do this and just using the movements alone to predict the behavior of the animal, this is what you get. So it's not very realistic. This basically shows you that you need the information from the brain to get realistic movements of the fly. Okay. So this is like a big surprise that we had. So when we actually used the data, that we had a training set here, and there was withheld data that we had that we never used to train. But the withheld data had these long pauses where the fly was doing nothing. In the training set, there were never these long pauses. But in the simulation of the downloaded fly, we can actually have these long pauses happen sometimes that look very realistic, like the real ones, but we never actually explicitly had it in the training set. So maybe there are some types of emergent properties. We actually had some other behaviors like the fly making round circles and things like that, running in circles that happen. Anyway, this is just a performance metric, as I said. And this is just to show you that we can actually do it the same thing. We can simulate for the whole brain and Zebrafish at single neuron resolution and get realistic motor neuron outputs at the whole brain scale at single neuron resolution. We can actually do this. And we can also do it for fMRI data of humans with doing a joystick task. So we might have the beginnings of doing basically arbitrarily downloading brains of things. So this method we call generative manifold networks, and is a new method that basically generates a network of manifolds that can simulate, based on real recorded brain data, realistic behaviors. Anyway, so with that, I'll leave my acknowledgments, and those are my collaborators. They're from the last few years, and I'll take questions.
Wow, thank you so much. That's totally fascinating. Does anyone in the audience have any questions?
Hello, I'm Markus Kaiser. I have lots of questions. So one part is, because you have the connectome for zebrafish as well, if you look at one particular part of the brain and look at your time series and you look at the dimensionality, so you look at the number of components for the time series that you're getting, does that correspond with the degree of connectivity? So let's say one part of the brain is connected to 20 other parts of the brain. Does it mean that there are 20 components?
So unfortunately, we don't yet have the connectome of the zebrafish right now. That's a few years away. People are working on it, but we don't have that. But what you actually have here is like the effective connectome for that particular behavior that you recorded in. So I think what we're picking up, if you look at Jeff Lichtman's recent things, they also tend to be like low dimensional. There are a few connections that you actually have on any given neuron. They have like synapses that have many contacts. They're really strong, supposedly, and a lot of them, they're super weak. So we seem to be picking up like the strong ones. And then we're not really picking up the weak ones very much most of the time.
Yeah. Okay, so that makes sense because they will drive activity less potentially, and
Exactly. And unless you observe it, then you don't. But
Yeah.
most of the times we see things are so the low dimensional ones are like the strong synapses. But if you have things like traveling waves where a whole bunch of neurons together drive one neuron, you tend to pick up all the ones that are coming together, you pick up as a single dimension because they are coordinated. So functionally they're one dimension, but in reality, there are many neurons together doing the same thing, but functionally they're one. So we identify those as one single dimension, although in reality, there are actually. So the thing is basically what you actually have is that we're reconstructing the overall behavior of the brain by having each manifold, which are basically all the decision-making components. We're not explicitly simulating the guys that are part of the bucket brigade.
Yes.
Yeah.
And basically for Zebrafish, you mentioned you have lower dimensionality for hypoxia. Could that just be due to higher synchronization for that state? So
Yes.
higher overall synchronization in the system?
Yes, it's more synchronization. It's also simplified the behavior. So before the zebrafish in default state, for example, is doing a whole bunch of different things.
Yes.
Once it wants to escape something, it only wants to escape. It doesn't do anything else.
Yeah, it's
So
only focus on one
dimensional
function.
reduction is
Yes.
basically the simplification of the behavior.
Yes. Okay, final question. So you have your time series, so you have the current state and then the previous one and then the one before that. And then you can use that to predict the number of components that are causing the time series, right? But that would also depend on your time resolutions. Let's say if you have EEG, you can measure with millisecond resolution or half a second or 1 second.
Yeah.
So you would
we're
get
limited by the
number.
data, yes.
So you would get different numbers of components for the different time resolutions, or
yes, potentially you can. That's the problem of having to use when you do the tokens embedding, you commit yourself to particular timescales in its original formulation of tokens. But in the second one that I showed, the generalized tokens, then 2011 extension, that one doesn't restrict you to a single timescale. So you can actually have multiple timescales where you vary the tau. The tau is no longer constant, and then you can actually account for any number of timescales. But when you actually have the real time series, so that's basically the tokens embedding is basically like an Eselsbrücke, is a stepping stone for you to get to where you want to go. And then it's basically once you identify the native variables, the real ones, those are contain every time scale that that neuron have up to the resolution of the recording. Obviously, you cannot get the stuff that you didn't record.
yes.
You actually have a measurement of, you can only get up to the level that you can measure. So that's a limitation that you cannot overcome computationally.
Okay, thanks.
We're pretty much at the hour, but to close it out, I guess I would wonder, what are the next steps for you in this work?
Right now, as I show in the slide, like Jack Gallant and Tianjiao Zhang, as like, for example, we're actually doing the download of the human brain and then trying to get humans playing video games simulated, and then hopefully the simulations will play video games in the way that the recorded humans do.
Amazing. We'll have to keep an eye on that.
Yeah.
Thank you, everyone. And thank you so much, Gerald, for joining us. It's been a total, absolute pleasure to have you on. So
thank you for having me.
Yes. And we'll see you all next month. Have a great day, guys.